Hello, everybody, and welcome to today's lecture on plotting in R. This is one of the best features of R, in my opinion, where the same way we've been coding and manipulating data is you can essentially build your own figures from code with really simple functions. Mostly, we're going to use ggplot. There are other versions that you could use, but I would say it's one of the most often used programs for plotting. And what the GG stands for is Grammar of Graphics. I think this is why this is so fun, or such an exciting part of R, is that you're really learning what is the basic grammar of how to produce a figure. And then you can use a lot of the same functions and other tricks we'll go through today to make just absolutely stunning figures in R, or plot your data in a very interpretable way. For today, what we really want to do is just the intro to this, where we're going to go over the layers that there are in ggplot, specifically ggplot2, which we'll load right now. Load these guys. And so really what we're looking at here is layers of information that we need to plot for a figure. So number one, of course, we're going to need data. That's probably pretty obvious. And then what are we going to do with that data is what we need to do is take that data and map it to some X, Y, or some sort of coordinate system. And that will be really important, and we'll spend a lot of time on this mapping layer here, which is also known as the AES layer, aesthetics layer. But let's, for now, we'll think of it as mapping. There's a stats layer where we can add more additional statistics as we go in our figure. Scales will allow us to see where we want our limits on our X and Y axes, things like that. Geometry is probably the third most important after data and mapping, because this is going to tell us what type of plot we want, or geometry. Do we want a dot plot, a bar plot, a histogram, a density plot? And a lot of these things are built-in functions into ggplot, making it very easy. And then the facet layer is going to allow us to make multiple figures in the same pane, for instance, where we can say we want to plot like 10 different DNA binding domains all in one figure. This can get unwieldy, where if you facet to too many objects, you'll have too many figures, and we'll get into that. Coordinates and theme are just kind of additional aspects that we'll touch on. We've made a theme for this class called Paper White, but we'll show you how you can load your own theme, which is going to say to R where to put the axes. How do you really want this to sort of come together as a thematic figure? And so let's start with the most important layer, data, which we absolutely need. And so in our class 10, where we did our data in ranges, we made this result file numpeaksdf.csv. And we can use read CSV to load that in so we can start plotting with it. So we're going to call this numpeaksdf, again, from the same as previous course, but we'll just quickly load this in. And there it is, and let's take a look at it. So this is all the DNA binding proteins we have here, their number of peaks, the total length that was covered. And we added this nice feature in our data in ranges, lesson 10, where we added the type of DNA binding domain it has. And we added these really handy features of what type of DNA binding domain it has, whether it has an AT hook, a BHLH. And we had a video previously that went over some of the ways DNA binding proteins have different domains that can bind to DNA in different ways. And so that's all marked in here now. And we finally have this column of whether the DNA binding protein is actually considered a transcription factor that helps activate transcription. Okay, so that's the information we have to plot with. And let's get started. So first, we're going to filter out any of these values that have an NA in them, and that we're going to use, feel free to look it up in the manual, is.na is a question. It's a logical question, so you'll get back true or false. And so what we're doing here is we're taking our numpeaksdf, we're piping it into filter, which we've seen before, And using this function isNA, where it'll say if there's an NA in a column or an entry, it'll say it's true. And we're going to do that in the TF column that we just saw. And this exclamation mark here means not. So we're going to say filter those that are not an NA in the TF thing here. So we can see our num-peaks TF right now is 460. And so if we do that, we see it went down to 438. So roughly 22 of the TFs didn't have an entry of whether they were or were not. So it was just NA for not applicable or not available. So let's look at what we got now. So we have 371 DNA binding proteins that are transcription factors and 67 that are not. And this is a little offset. There's quite a 5-fold more that are yeses, but for today's purposes, this will help us plot different features of whether data has a relationship to whether it is a TF or not. And we'll get into that in a second. But let's go back and let's start with the very, very, very basics is the data. So we're going to call ggplot. That's going to do the plotting for us. That's the function we're running. And what it first wants is data. And so we're saying here, we're take our num-peaks df, and that's the data we're going to be talking about. And so in this layer here, it's going to now know everything that's in that file. So it's going to open it up, and so you don't have to index into it or anything. It's well aware of the file that where your data is. And now this AES layer is that AES or static or mapping layer is really important. And so what we're saying here is after we say what the data is, we have to give a second argument of what the mapping is. That's the absolute minimum you're going to need in the first layer here. And so let's just say X equals the number of peaks and Y equals the total peak length. Doing there is we're saying, okay, here's where all the data, here's where the data is. This is what I want to map in that data frame. And then this plus sign is going to be really important as it's going to indicate that we're going to another layer in the figure. So I mentioned the first layer is data and then mapping is what we've done here, but we need a geometry layer too, a third layer. So we're just going to tell it to make a plot with just dots with the values of X and Y. So if we see this here, we'll run it and we get the plot we saw in the R data and ranges here, where we have this one outlier here at about 150,000 peaks, and whereas most have about 50,000 or less. Okay, so that's great. And now you've often seen figures colored in interesting ways, and this is actually a subtle but important point, and we'll outline it here with these two examples. So really we're doing the same thing here. We're saying ggplot, look at this data frame, and now we're setting the axes the same. But let's say we want to add some flair to this figure and color the points purple and turn them from circles into squares. Because that aspect of color doesn't depend on the data, we don't have to put it in this layer. We'll do it after the first layer into the second layer of the geometry of the point dot plot, and we'll add it there. And you'll, for now, just bear with me and we'll see why that becomes important later, that we don't do the coloring in here unless it depends on data inside this data frame. Okay, so now if we run this, we've just added the same thing we ran here, but we added some information in the geometry layer. And each layer has many different aspects you can add to it, and you can just add a question mark this and see what parameters you could put inside here. And so for now, we'll just do the simple example of a shape and color. So when we do this, cool. We just sort of coerce the data to become squares, purple squares. So that really doesn't give us any new information. It's just something you might prefer as a color thing. But now let's say we did want to color these dots by the data, whether it's a transcription factor or not. So we had the 371 that were and 60 some that weren't. And so what we can do now, we'll see here we have our data, our X and Y are set. But now we added another feature in the AES layer, or the mapping layer, where color equals TF. So this is technically a function where R is going into the data frame and saying, okay, if you're a transcription factor, I'm going to call you one color if your answer is yes, and another color if it's not. And R will default to its own colors. And again, we could add more information here onto what colors we wanted, but for simplicity, we'll do that. And so after we have all the data mapped and then now colored by the, whether it's a transcription or factor or not, we can just add a new layer as the dot plot, or these plots we've been looking at here. So let's look at the difference, what we get. Okay, so now we can see there's two different types of colors. And we got a legend for free, to be honest. This is one of the features that ggplot is so smart. They said, okay, in color, there was two values, or in TF, there was two values, yes and no. And we could have left NA in there and we would have had a third color. And so R knew that automatically and just defaulted to these two colors. And we can see that our outlier out here is a transcription factor, and that really the non-transcription factors and the transcription factors follow very similar trends below 50,000 peaks, but above 50,000 peaks, they tend to all be transcription factors. Okay, so that's where we need the data to color. And so that's where we would add color in the aesthetics layer and not in the geom layer, which we just did above. Okay, so let's look at these DNA binding domains. There was several different types in there, and it's kind of confusing to scroll through that big long table, but let's just table them where we're going to look into the data frame, into index DBD, and see what we have here. Okay, so we can see all the different types, and there's several in there. But let's look for those that have, some of these only have like one entry, like P53 only has pipsqueak. Okay, it has two entries. I've never heard of that. But we really don't want things that only have one value. So let's look at what the more abundant DNA binding proteins that have at least 100 examples of a single DNA binding domain. That will, DBP and DBD sound very similar and a little bit confusing, but will make sense that we have 438 DBPs, and each of those 483 has one of 15 or so different DNA binding domains. But if we look to see which ones have more than 100 entries, just using table here and indexing and filtering by greater than 100, we can then see which ones those are with names we learned previously. And so what we can see is this C2H2ZF has more than 100 entries, and for today's purposes is a good starting point to just, we'll take a look at that specific DNA binding domain. So now let's say we want to plot, same thing we've done before, num-peaks-df is our data, our X and Y axes are the same, but as we just colored by TF up above, we can actually run a function in here. And I'm just giving you the most simple function for now, but you could do whatever you want in here to run R functions on the data frame as long as you're in this data layer. So what we're doing here is we're not just coloring by DBD. That would give us 15 different colors, but we're saying we're going to color by DBD that is absolutely equal to this category. And so that will simplify it to just back down to 2 colors, and then we can pipe that or add the next layer of a point as we have been doing. And so we do this, what we can see is, okay, yeah, there's a lot of these C2H2 zinc fingers in our data, and we can see that the highest number of peaks in genome coverage is due to one of these C2H2 zinc fingers, whereas they're kind of intermixed, as we saw, very sort of non-differential down here. But we can tell that our top top DNA binding protein is a C2H2 zinc finger DNA binding domain. Okay, so that's kind of nice, but there might have been a difference that maybe all the orange dots were up here. In our case, we didn't really see much of a separation by this category, but you can see how other categories could be more informative in separating the data. So let's take a look from the simple XY plot to a histogram, which will only have the x-axis. And then the geom histogram is going to automatically count the number of features in each bin of the x-axis you give it. So we're just going to say number of peaks for now is our x, and make it a histogram. Let's see what that looks like. Okay, cool. So we have a histogram where it defaulted to, I don't know how many bins it defaulted to, but it said, you know, there's more data in here than you're giving yourself credit for. Why don't you add more bins? And what bins is just simply is how many vertical lines we're going to have. So if we had one bin, we would just see all the data and it would be meaningless. If we had two bins, we'd see a bin somewhere here and then one over here. The more bins we add, the higher and higher the granularity we'll get. Okay, so let's do that. And let's see, just for dramatic sake here, let's add this to 300 and see what the difference looks like. And you can see it looks much more refined, a little bit harder to read. I think 300 is too much. It suggested 30. So let's go back to that. And what we've done here is we've just simply said, here's our x-axis, here's our data, here's our x-axis. And then now we're adding features in the geometry layer. So we are out of the data and mapping layer into the geometry, and we can customize our plots. So let's rerun this with 30. Okay, back to something looks fairly similar. Distribution, really this is the same as the x-axis values in the XY plot. Okay, so now let's manipulate the data layer here with a histogram. So we have our data, our aesthetics, we're saying the x to be number of peaks still, but now let's fill it by TF as we did before with the XY plot and a histogram with 30 bins. So we got. Okay, so cool. So fill here allowed us to say in the data, because it depends on the data, we had to do it up here, that we're going to fill by whether it's a TF and yes or no are the only values in that column. And it plotted it as such. But it's kind of hard to see because the two distributions are overlaid, and that's not a very nice looking thing. So we can add another feature inside of our geom histogram layer where we can add position. And again, you can run the manual on these and see all the different types of things you can put in this layer separated by commas to indicate a different function. So position equals dodge will allow the bars to never overlap. And so when we run that, we can see the plot is a little easier to read, and we can see the differences in numbers in the distributions a little better. An even better way to see these sort of plots, like where you have two very similar distributions, is a density plot. So now we can just add geom density to this exact same first line and see what it looks like. We're filling by TF, but we're going to make it a density plot. And again, okay, so now we're at the same problem where we can't see them very well, but density will allow us, well, first I should mention that on the x-axis, we have number of peaks as we indicated, but R is going to default, or ggplot is going to default to know what a density plot is telling you where is most of your data. So if you took the integral of all the area under this blue curve, it would equal 1 in a density plot. And same for the orange one, it'll equal 1. So you can see these axes often look funny, like what is 2e to the negative 05? Well, that's a very small number, but that's only one data point. And so you can see as you go, you're adding that cumulatively and up until the area under the curve is equal to 1. Okay, so let's say we want to see a little better underneath here. And so we, in our geom density, we can start to add features too. So we can add alpha, which is sort of the opacity, if you will. We'll set it to 10%. whereas right now it's defaulted at 1. And okay, so we can see a little better between there, but the two black lines are kind of confusing now and can't really tell the colors as well. So let's add, let's change the color of these lines, whether they're a TF or not as well, instead of just the underlying data. So the way we can do that is we have our data, our axis is the number of peaks on the x-axis, our fill is by TF, and now our color is also by TF. So we filled the plots before with the colors, and now the color is going to be the line here. We'll make that is the same, whether it's a TF or not. I changed the alpha to 0.3 because we'll see. Oops, missed a G. Now we can see the blue line and the orange line, or salmon line, I should say. Okay, but what if we wanted to get rid of this is kind of garish, and so really the cleaner version of this would be to set the alpha. This is all the same, setting the alpha to 0, and we just simply have the two lines that tell us whether it's a TF or not, and really we can see they follow fairly similar distributions along the way. So most of the data is down here, and less and less and less of it is out here. As we've seen. Okay, so now what if we want to add another layer? Can we just add layers? Can we add a point to a density plot? The answer is yes, if it's logical, that is. And so let's go back to our XY plot here, where we have our x-axis is number of peaks, y-axis is total peak length as we did, and we did geom point before. So basically, if we just ran this up until this plus before we add that next line, we'll get the same plot we got in the beginning. But now let's add another geometry layer, the 2D density map. And this is like a topographic map you might have seen for how tall mountains are or other features. And so when we run this, we're going to get both of these here. So we can now see where the density of our data is. Most of it's down here. And as we move out, these are the different sort of density quartiles of what data is fitting within the density, and all of these are sort of outliers outside the main density of the data. So that's kind of handy because some of these black dots start to look very overlaid, but you can now see the peak of the data, or the peak of the mountain is here, and you sort of go downhill as you go here. So this would be the trailhead, and then you would come to the climax of the mountain here. So that's kind of nice, but does the order matter? It does, and this is going to be very subtle, and you can do it on your own. But if you look at one of these dots where the line is going over it, what we did first was a point, the dot plot, and then we added this. So now the blue line you'll see crosses over the black dot. So it's on top of it because it was added second. If we just simply flip that, this is all the same. If we just flip the order of those two around, we can see this. And now the black dot is above the line because we plotted the points after the density, or at least that's the way GG plot thought of it. So order does matter. It's pretty subtle in this case, but it can be more dramatic in others. So let's say we wanted a linear line. You always see these sort of lines saying, oh, our data is following this trend. And this looks very linear. It looks like we could, a slope would be y equals mx plus b, where m is the slope. And it looks pretty much like 1, where we get one value on the y-axis for every value on the x-axis. So we might think it's 1. And we can actually add that. That's called a geom AB line or AB line. You can look that up in the manual of all the different things you can add to it, but you have to add slope and intercept. This can actually become a function of your data, a regression. We'll get into that stuff in more advanced R plotting. But for now, this is just a way you can add a third sort of feature to your data. I'm going to take off this 2D density for the sake of simplicity. And so what we have here is we're reading it. There's our data. Our X is number of peaks, Y total peak length, same as we have been. Let's make it the same plot. But instead of adding a 2D density here, or a geom density, let's add an AB line of a slope of 1,000. You'll see y in a second, and an intercept of 0. Okay, So in here, you can, in the manual, see what features can go in here, but these two are going to be required, slope and intercept, for this layer feature. Okay, cool. So now we can see our data is quite linear, that most of this data doesn't fall very far from this straight line. But why is the slope 1,000? And let's see if we did that at 1, which is what I would have guessed just by looking at it. It should be a slope of 1. But that's not. So what it's saying is that the slope of 1 is so far down below, it almost looks flat. Whereas if we do 1,000, we see it fits better with our data. And so this is a manual way of playing with the regression to say, like, oh, wow. And so what does that mean? Why even have this example in here? Well, it's kind of interesting that if you played with this, you could keep guessing different numbers, and it's pretty quick, easy to get to where you want to go. When you see 1 is flat, you can add it to 100 and you'd see it somewhere around here, add it to 1,000 and it's perfectly linear. And we want the y-intercept to be 0. And so what that means is each peak is about 1,000 base pairs, or 1 kilobase, which means that each time we step 1 peak, we have to go up 1,000, 2 peaks, up another 1,000, 3 peaks, up another 1,000. That's a function of this large scale here, being auto-scaled by ggplot. Just a nice way to add other features to show your data is linear. You can make this a dashed line with adding other features. You just add commas, and you can really tailor all the layers to your liking. Okay, bar plots, we'll look at that real quick. There's only one real thing to think about. Bar plots are pretty simple. We have our data, our x-axis is, let's say, the types of DNA binding domains there are. It's probably the easiest way. We should have looked at it in the beginning. And we'll add geom bar for bar plot. Run this quick. And we can see here all the different entries in that DNA binding domain column. And we see C2H2 zinc finger as being the highest. Remember, we said what's above 100, and we only got that 1 out. Unknown has about 60 in there. And homeodomain has a few. So really, we nailed the big one here. And that's just a standard bar plot. And what happened there is ggplot said, okay, for each DNA binding domain entry name, I'm going to go count how many there are. We've seen that in summary and table before. So that's what it's doing by default. But let's say you wanted to set the y-axis equal to something. Let's say we wanted to see the bar plot for the number of peaks for all 438 DNA binding proteins. So what we would do there is we have our data frame, our data aesthetics layer. I just typed this a little different so you can figure out your favorite syntax. But this shows that the X is going to be dBp. And it's important to note it in a bar plot, like, why would you have a y-axis? That it should just be the counts from that identity. And so if we set a y-axis here in the data, because it will depend on the data when the bar plot goes to make it, we can now add our geom bar graph. And inside there, the parameter we're adding is stat equals identity. And that means the statistic it's going to look for is the identity in the y-axis, which is the number of peaks. And so the x-axis is now going to have every single dBp, or DNA binding protein, all 438 of them. And we're going to tell it to force it to tell us the number of peaks for each of them in a bar plot. And so that would not happen if you did not do this. You would get an error. Let's run it the correct way. Possible to read, but we can now see that you can set your own y-axis with stat identity. Try deleting that and leaving it all the same and reading the error as an exercise. Okay, so we've gone through the most important aspects so far. The data frame kind of stays the same most of the time. And knowing the AES layer versus the geom layer is pretty good. And so if you need to color something or manipulate something that depends on the data, it has to be in this mapping layer. Otherwise, you can customize your plots in the geometry layer as much as you want. Okay, so now let's look at scales. And that's really just about how big and small things you want. The most obvious of this is how tall or you want your y-axis, or what the limits of the x and y-axis are. And we'll do that in 1 second. But first, I want to show you what really is going on with this scale x continuous, scale y continuous, and also scale color. This is less important, but I'm just adding it here to show you that this scale layer has a lot of features you can keep adding, just like we added multiple geometries with the point plot and then the 2D density. We can add different scale layers as well. So scale x continuous has lots of different parameters we can add in here, but essentially what we're doing, if you add the breaks, is going to say, tell me where to make the tick marks on the x-axis. And that can be important if your data is spread out over big numbers like ours is. Scale y continuous, one function in there is transformation, or trans. And we want our y-axis to be log 10 because the genome space covered is 1,000 bases for every one peak. So that would at least change it down to log 10.3. And make the axis more readable than those 1e to the numbers. And this, let's just think about as you can have ggplot scale your data so it's more visually visible, the differences in the data. And this is palettes. You can set it to that. You could just leave this blank and it'll default to a palette it wants, or you can change the number on these any which way you want. Let's just do 1, and then you can try others as you go. So when we do this, we can see only just by adding scale layers. So we've done our data, our mapping, same as we've done before. And now we've added the scale functions to sort of just add a little bit of features to our axes. Let's see. Okay. And this is where I was saying is that you can see that if we just add like, 1,000, 10,000, 100, 1,000, 10,000, 100,000, a million, that our data is not evenly distributed in a linear way. So we can see a lot of the data is here. And even notice that we added on the x-axis 1 million, but it didn't print the 1e to the negative 6 here, or 1e to the 6. And that's because this data point is less than 1 million. And so it will only plot the breaks that are relevant to show you data that you have more than. And then on the y, we can see it's a little more readable with 5, 6, 7, 8 being log 10 and easy to read. So we can see that like most of our data is down here and very little of it is out past 100,000. And the scale was that color. We could have picked 6 instead and gotten sort of a black and white figure here. Okay, so that's just really, sometimes I like just seeing that the data is very loggish and pointing that out, that these are really extreme outliers and not just sort of linear steps out that XY plot sort of made us think. And really the most obvious thing you're going to do here is X limbs and Y limbs, where you're going to set the limit to the X and the limit of the Y. So let's say we wanted to zoom in, where we're going to zoom in on the X-axis only between 250 peaks, number of peaks, because that's where we made this list from, more than 250. The DNA binding protein had to have more than 250 peaks to be considered. And let's go up to 2,000. And so with the Y limits, we can then keep it this sort of in between here, because we're going to go way down in this range. And so we're basically zooming into this corner by simply setting X limb and Y limb. Again, this is in the scales layer where we've done our data. We've mapped it. We've even mapped the color of the TF in there, made it a point. And now we're just changing the limits of the axes. And so you can see it's not going to plot the whole thing anymore, but now we're only in this range here from 500 to 2,000. and we can see the total length of the genome covered by those peaks. So sometimes you might want to make this plot and have like a zoom out window of this plot, for instance. Okay, so that was great. We're really see we can make a lot of different plots pretty fast. Now this last couple things, this one's tricky. I'm just going to be honest. Facets are really a powerful feature of ggplot, where there's facet grid and facet wrap. We'll go through a simple example of both of those. But you have to be really careful here, because what R is going to try and, or ggplot is going to try and do, is make as many figures as you facet. And so facet just means it's going to go into a column, and let's say TF, there's yes and no. So it's going to give you, it's going to give you two different plots, the plots of the yes and the plots of the no, if we faceted on TF. And facet wrap is going to test multiple combinations. And so we'll first start with facet grid. And you can go through this more if you want, but this is just to make this exercise a little more reasonable, is if we index into our DBDs, which we know there's 15 different types of DBDs, let's find some that have, let's order them so that we know how many peaks they have. So we've indexed into there, and then we're running a sort of a function in that index, if you will. And so we can see again that our top one is here, is C2H2 of ZF, unknown. Yeah, let's skip it. BZIP would be the second one, and homeodomain, very cool. It's a very cool. Hox genes have homeodomains, and they pattern most of the body. So it's a pretty cool thing. So let's take those three now. Whereas before we just looked at this one, this is where facet becomes handy. We can make a plot for all three of these just by faceting them. Okay, so let's make a simple object, which is just the names of those three DNA binding domains here. Okay, so now we have that value here. It's just simply the character list. And now let's do a big function, or not a big function, but let's show the power of ggplot here. So we've given it our data, that's fine. And now, not only have we given it our data, what we're gonna do before we give it the data is filter it. So we're gonna take the data, filter it, and what the filter is gonna be, we're gonna go into the DBD column and look for all the DBD names that are in our DBDS. Probably poor nomenclature there, but it's just gonna look for those that match these three names. So that's kind of cool. We already are filtering and doing functions in a figure. So we can quickly use the same functions we were using to generate this data table inside the data layer itself. And now we move to the AES layer. So you can see now our data thing is not just numpexdf, but we can run all those string extract, unfilter, count, which max, all these functions can be run on the data before sending it to mapping. Okay, so now mapping, we're doing the same thing. X is number of peaks, Y is total peak length. And now here's where we're adding the facet layer. We're adding the plus sign to add one new thing, which is facet grid. Is easier to understand for now because it keeps the X, it's gonna always keep the X axis the same. And this is tilde we're gonna see for the first time here, which simply means pass to, essentially. So what facet grid is gonna do is look in the DNA binding domain column, and then that's been filtered to only these three now. And it's gonna say, which of those is a TF? I wanna know, give me a figure for each of these three domains and tell me whether it's a TF, yes or no. Okay, and so it's gonna do that automatically. So facet grid is pretty simple, and we'll see why facet wrap becomes a little more flexible. Then we'll simply just add the geom point here. Okay, is our geometry layer. So we've got this nice data filter function, our mapping function, and now our faceting. We're going to go into the three DNA binding domains we looked at and say, how many of those are TFs, yes or no? And so we'll get back how many, try and guess how many plots we get back before this plots. Okay, we got 6 because Facet Grid is going to ask only these questions. It's going to ask it no matter what, even if there's no data. So what we can see here is for the beta zip that we wanted to look at, they're all transcription factors. So there's no dot here. But Facet Grid will still plot it because it wants to keep the x-axis the same between the 2 aspects you just faceted out. I know this is a little conceptually difficult, and we're not going to get into it much later in the class, but I think it's important just to highlight some of these simple examples. So now C2H2 zinc finger did have one DNA binding protein that had this domain was not considered a transcription factor. Same with homeodomain. There was one out here. Had very few peaks, as you know. Notice that these 2 dots are out here. So this is where facet wrap would come in to play, is that we don't really need this plot because we don't need to know that there was nothing in there. And so let's run the same thing here with facet wrap. Okay, so there's markdown here to walk you through this, but this is what we just ran here. And now this is the same thing, same faceting, but we're just changing it to facet wrap instead of facet grid. Let's see what the difference there is. Okay, so what we can see now is it's still doing the same thing. It's saying, okay, I'm going to go into the 3 DNA binding domains you told me to look for, and then I'm going to plot whether it's a transcription factor, yes or no. And so homeodomain did have that 1 dot out here, so it still gave us 2 plots. Same with C2H2 zinc finger. Most of them are transcription factors, but there's that 1 guy that's not. And now with beta zip, facet wrap was a little bit smarter, and it was like, well, there's no data in the no column, so I'm just going to say yes, as the reader should know that there was no, if that makes sense. So you don't necessarily have to fully understand the difference between facet grid and facet wrap, but I think it's important to see how quickly you can make many figures. And we could have made all 15 DNA binding domains. But then this gets unwieldy, and you might as well just make each figure separately and put them together in Illustrator. And one other thing is that you don't have to facet wrap on everything, or you don't have to have 2 values here. So this is just a short example to show that with the same setup here, same setup is right here, except for we're not doing DBD passing it to TF. We're saying take TF and pass it to nothing. So really we're just going to facet wrap whether the yes or no and plot that for our number of peaks and total peak length. So this is a different plot. This is that XY plot we made. And now we're going to change it here to just facet by TF. There's markdown here to tell you more about that. And so what we can see is TF only has 2 variables in that column, yes and no. And so what we can easily do, and this is probably the most common way you would use facet, is just to say, all right, I want to know what the data looks like, whether you are or not. We also saw that by changing the color, we could have done the same thing, but sometimes you might want the plots simply next to each other. And I think that shows the power of facet, that you'll just get them right away. Now, this is defaulted to 1 row and 2 columns of figures, but you might want to see them in a different orientation where they're maybe on top of each other. So in facet wrap, we can, as every time we add a comma, we're essentially adding a new feature to this layer. And so everything is the same here. We're just adding facet wrap TF to itself, to the yes and no's in there. But let's make it n row equals 2, whereas right now we only have 1 row. Okay, let's see. So we have 2 rows, and now we can see they're plotted on top of each other. And there's other features you can get into in facet wrap, but this might be like wild type versus knockout. It might be much more interesting situations than just simply yes or no for a transcription factor. Not that it's not important to know the difference, but This will allow you to sort of allow ggplot to go into your column and make both plots for you, separating the features in that column. Now, if that column has 100 features, then you're going to have 100 plots, and it's going to get unwieldy. And then just to show that you can add more, here is we can also scale these so the scales are free. Right now, it's keeping them the same. And so we can see the no is kind of more dampened down here. But if we wanted to make them scale so that we're scaled by the data in this one, and then this one would be scaled independently, we can say scales equals free. This is the same as before, but instead of just showing you, instead of n row, you can also say number of columns. So if we say one column, that's really the same as what we did here, is making 2 rows. And so when we run this, all we're going to see is now the X and Y axes are a little different, and it lets you see the texture of the node data a little bit better than where it was squished by being defaulted to the biggest Y axis here. Okay, so that is Facet. Coordinates are almost always for this class are going to be Cartesian coordinates, or x, y, or x and some value. But just for the sake of touching on each of these layers, let's look at this where we have number of peaks df as our data. We're going to make a bar plot, so we only need the x-axis for the types of DNA binding domains that we've been looking at. We can make it a bar plot, but we can add another layer here called a coordinate polar. So the coordinate layer is going to say, where do you want to put this space? And we're looking in 2-dimensional space. We might want to be in 3-dimensional space. We might want to be in, with Fourier transform, you might want different sort of plots. So let's just see what this looks like with polar coordinates, which really are going to say, like, make a full circle for 100%, and then subcircles for that. So really, you can see it puts like a clock on here, and then the width and length of this bar is the number of them there are. And so again, we see C2H2 zinc fingers as the biggest version. I mean, we could have saw this in the bar plot easier, but if your data is really hefty and there's one outlier, this is a good way to see it pointing out there. And unknown we saw was the second biggest category. You had homeodomain. So these little bars are pointing to where the most features are in the data as a circle. Now, this is harder to read than a simple linear bar plot. But just to show you that you can change the coordinate space. And then again, in any layer, you can add different features. You just look in the manual and see what are the possible things I can do here. And I can have the polar coordinates, instead of going in this like clock-like fashion, we can orient them based on their theta, or the angle that this bar is at. So this is a bar plot, but it's just in polar coordinates. And so here's our Z2H2 zinc fingers, where we know 150 is about the max, and this is what that circle represents. This is probably unknown with about 60. This is probably homeodomain somewhere in here. It's really hard to read. It is nice to know that you can zoom in here. Sometimes that helps, sometimes it doesn't. But it's a nice little feature on the pane. Okay, so theme is the last aspect, and for class here in our setup R, we called it paper white. Oh, here it is, theme paper white, and it's a function. It's not that bad, but it is, you don't want to make all this code every time you write a figure. And so really what we're saying is we want the background to be white, we want the figure legends to be this size, we want the color to be that. So every time we put in a theme of paper white, all of these things will now be included. And you can see that this could be annoying to have to add all of these to each of those ggplot. So ggplot also has a bunch of themes built into it. Paper white is just something we like. And you can see that was added here. This is from one of our other figures in 10, our data in ranges, but we had theme paper white here. We didn't really go over it then. You can see how simple it is now. Now it's like those functions we wrote in our intersect functions or plotting functions. Once you write a big chunk of code that could be useful, it's good to bundle it into one of these .r files so that you don't have to do that every single time. Okay, so when we look at this here, we're just saying theme paper white, but I thought it'd be nice for you guys to see, I mean, this will run, and we're adding the idea here, you can go over it, is that we can add an X label, Y label, and a title, subtitle. There's all kinds of features you can add in here on top of the Paperwhite theme. So the Paperwhite theme can give you the real hardcore fundamental way you want your colors to look, like gray and black and white out here, the type of fonts on the things, but you still might want to change the names of the axes. So you don't want your theme to always have the same names of things. So we did have to add the labels, even though we had a predetermined theme here. And you can look at all the different themes in R. It's probably better to Google, or ggplot. It's better to Google ggplot2 themes. Cleveland turns out to be a theme. So here we're just doing num peaks df. X-axis is number of peaks. It's a density plot, and we want the theme Cleveland. So we made this exact same plot up above, or we can just do it here by not adding that layer. And that's our nice density plot. And then ggplot's going to add these nice little features that are in Cleveland, if that's what you prefer, which are tick marks and some other subtle features in there. So depending on what you want, it's good to know a theme, and it helps save a lot of time in writing lines of code to make it look just perfect. Okay, so there is your introduction to plotting in R. I know it seems like a lot, and we're going to do this more and more, but I think the key things to take home today are you need your data. You can do functions on that data layer. If you want to color something that depends on the data, it should be in the mapping or aesthetics layer. You can run functions in the mapping and aesthetics layer. And then you need a geometry, what type of plot you want. And those are the main sort of take-home points, but we wanted to go through to be for the introductory lecture, and feel free to go back and look at some of the markdown in here, really getting you to understand these layers. And they don't necessarily have to come in order, but these are the sort of fundamental buildups to the ultimate figure. So with that, we have a class exercise to go back to 10 R data ranges. This is an example of one there, and just sort of take a look at what was going on now that you have an understanding of that. The last thing I should say is that we could do here, we could do gg save and say class figure dot PDF or whatever we want it to be, and it will automatically save this. I don't want to do this in our working directory right now. The other way to do it is simply do this. You can export, save as PDF, save as image, copy it to your clipboard. You want to make lab meeting real quick, you can just copy these and paste them into your Keynote or PowerPoint, however you prefer. So that is it for plotting. It's one of my favorite features of R, how you can quickly scan data and start to understand results. And with that, thank you, and we'll see you in the next class.